this computer. There we go. Okay, so I did one of these back before Christmas on looking at trends in species, and there seemed to be some pull to do something else. So one of the things that I've been interested in for a very long time is species distribution modelling. So that's tonight's topic, but it's a bit of a big topic. So what we're proposing is to split it into 3 talks at fortnightly intervals. So tonight I'll try and cover a general intro, what's involved, and run through a bit of a demo. And then in 2 weeks' time, the plan is to try and talk a bit more about what are the problems, what you can do to mitigate them, and sort of tuning and improving the whole system. And then the final one, look at the possibilities of forecasting. That one might be a bit shorter, I think. But anyway, we'll see how it goes. See what interest there is. So probably the thing to do is share my screen and get going. Really. Okay. I need to optimize for video, so we can do. So can everybody see that if I go to full screen?
Yeah, that's fine, Stuart.
It's fine.
Good, good. Okay, so species distribution modelling. What are we trying to actually do? Well, I mean, in a British context, it's pretty clear that the geographical sampling of the Hoverfly Recording Scheme is pretty biased. I mean, this animated GIF shows the density of recording in 10-year intervals. So each frame is a subsequent 10-year interval running from 1980 up to 2022. And you can see pretty much all the time down in the southeast London area is the most dense, which is probably not surprising because that's where everybody lives. There are also other high density areas, people like Sorby naturalists in the 80s and 90s, the Gloucester local scheme, very period when Roger lived in Stamford, things like that you can see popping up. But if you look in central Wales or in northern Scotland, you'll see it's pretty low pretty much all the time. So we don't get an enormous amount of recording from the uplands, and particularly the far northwest, the Highlands and Islands of northern Scotland. So if we look at a species, even a fairly northwesterly distributed species like Cryptotoxinum arcuatum there, we really have to ask the question, is how realistic is that distribution? And for example, is this area up here actually an area where it isn't particularly common, or is it just totally under-recorded in that area? So it's a bit difficult. So one of the possible ways of filling in the gaps is to look at a species distribution model. And that's some sort of a go at the distribution of Cryptotoxinum arcuatum, as species distribution models would tell me it is. So the sort of uses these things have had, if you start looking at the literature and so on, by far the biggest one is filling in the gaps, and clearly that's a very big problem in parts of the world which are much more poorly recorded than we are in the British Isles. So if you're looking at Australia or South America, or quite a lot of Africa, for example, for a lot of species, the knowledge of distribution is extremely sparse. It's down to a few papers, a few specimens in museums and so on. So if you've actually got to come up with conservation policy or that sort of thing, you've got really big questions on where do these species really occur and so on. I mean, a friend of mine ended up going and living in Australia, and he was interested in staphylinids and found that whenever he turned over a stone, he could find new species. And it's a worrying statistic that there are more species currently in museums in Australia awaiting description than the current entomologist could handle in a century of work. So, in those parts of the world, you just don't have the recording effort. So you really haven't got much else to do. It's also been used with invasive species. So if you get something coming in like maybe the ash dieback disease into Britain, can you actually come up with some sort of a species distribution model that will tell you where it's likely to spread to, which perhaps gives you some clue as to where to put your efforts in actually trying to control it? Or more generally in conservation planning, where best to direct limited resources. I mean, I got involved when I was still working with butterfly conservation, who wanted to survey checkered skipper and I think the other one was small pearl bordered fritillary up the west coast of Scotland. And clearly they got fairly limited effort. And so they wanted to develop a model to best give them a clue as to where to spend that effort. I did actually manage to persuade them to include some squares that the model predicted as fairly low, so we could actually use the results to test the model, and it worked remarkably well, actually. And of course, as we've already mentioned, one of the things you're seeing quite a lot now, and it comes up quite a bit in papers, Roger and I went to a symposium in France last September, and there were about 3-4 posters about predicting distribution under climate change from that. So it's clearly something people are trying to do. If you can fit a species distribution model and fit in forecasts of how things might change, then you can possibly get some clue as to how species might react. And that again informs your actions, where you put resources and so on. So the basic assumption behind it really is that species have a habitat. And you can define a habitat as the array of resources, both physical and biotic, that need to be present in an area to support the survival and reproduction of the species. And if you can quantify these physical and biotic factors, then there's the possibility you can use the knowledge of the distribution of those resources to predict where the species might be expected to occur. In the past, that was remarkably difficult to do because you try getting information about what the rainfall is and that sort of thing before the advent of satellites and sort of global coverage of these sorts of things. But certainly over the last 3 or 4 decades, since really about the 1970s, with increasing coverage of satellites, this sort of information has begun to be available. Comprehensive, high-resolution, global coverage of all sorts of things, stuff like topography and land cover and temperature, rainfall, and so on. And so it's now more possible to try and do this sort of thing. So what we're talking about doing is taking the known distribution of a species, the places we know it occurs, taking what I'm going to call environmental layers, which are essentially maps of various things that might be of some importance to that species. And there you can see things like mean altitude, weather, which I think in that case is annual rainfall, and the dominant soil class and so on, and try to produce a model which essentially is doing a pattern matching job. It's saying, at those points where we know the species occur, what sort of pattern of those variables are specific to those points. And if we can do that successfully, we can apply that model to the full set of environmental layers and get a potential distribution, some sort of idea of where else those conditions apply, and therefore where else we might expect the species to potentially appear. And it's worth pointing out at this point, there is a big difference between the potential distribution of a species and its actual distribution, in that species don't necessarily occupy the whole area. They could potentially provide them with the right conditions. And there are a number of reasons for that, the obvious ones being geographic barriers that stop them distributing to parts of that potential range, or the presence of other species, competitors, whatever, that are keeping them out. So, for example, I ran some models on a Northwest European basis a little while ago. And one of the things that came out of that is you get a pretty good fitting model for Eristalis rupium. And if you apply that model across Northwest Europe, it suggests it ought to occur across Ireland. And given its northwest distribution in Britain, that's no great surprise. But so far, at least, Eristalis rupium has not been recorded in Ireland, so why not? And, you know, the obvious thing to think is that, a bit like snakes, maybe at the end of the last ice age, it didn't occupy the British Isles before the Irish Sea was formed, and therefore been unable to cross the Irish Sea, and so it hasn't got there. I mean, another one you might think about is that through most of the last century, red kite was confined to Wales, and clearly we now know that the most of the British Isles was very suitable for it. What kept it out? Well, it was us. It was human persecution, and with a little bit of help, it's managing to repopulate that range, which was obviously eminently suitable for it. So its potential distribution was much wider than its actual distribution. And I think this is a pretty common thing. So getting a species distribution model can potentially tell you about the potential distribution of a species, but you need to take quite a few other things into account if you're going to start looking at the actual distribution. So the process, really, there's 4 steps. We need to know known points of occurrence of the species. We need a suitable set of environmental layers that are pertinent to the biology of that species. We need to train some sort of a model. And then finally, we need to work out whether that model's any good or not. We need to evaluate the model. As far as Hoverfly Recording Scheme goes, I've tended to use 1 kilometre square grid cells from which the species has been observed in a given time period. So that, for example, is Chrysotoxinum arcuatum 2010 to 2022. That shows where the 1 km squares are. Obviously, I've had to make them a lot bigger so you can see them. If I actually plotted 1 km squares, they'd be too small for you to actually see them. So I've plotted bigger blobs over them. But you can see where that species has been recorded according to the Hoverfly Recording Scheme in that period. Then we need environmental layers. And these, as far as I've used them, are 1 by 1 kilometre square raster maps for Great Britain. Now, what do we mean by raster maps? There are 2 ways, really, of storing map information termed rasters and vectors. And that gives you a reasonably good sort of look at what the difference are. These are both taken from Google Maps, and on the left, you can see where I live, where I'm talking from now, which is 255 Eastfield Road here. on Google Maps, and here is Google Maps with the satellite layer turned on. And again, you can see my house here with the solar panels 255 on it. Now, these are obviously different ways of storing the same data. This is a vector map, and you can probably get the idea that Eastfield Road is stored as a couple of lines designating the sides of it. My house is approximated by a rectangle, which will be stored as probably 4 coordinate points. So this is stored by storing points and joining them with lines. It doesn't take a lot of storage space. It's a very economic way of storing the data. And if you're doing things like calculating distances or working out a route, this is the ideal way you want the data. On the other hand, this satellite map is a picture taken by a satellite. It's an array of dots, and each of the dots is stored as the colour of that dot. The two have been scaled, so they're equal, and this shows a lot more information in some senses. You can see where all the trees are. You can see things like the fact my house has got solar panels on the roof, and the little wing off there also has solar panels. You can see one or two of my neighbours have got solar panels. You can see another house there with a whole load of solar panels and this block of flats with solar panels on it here. And clearly that information isn't on this map here. But if you actually wanted to do something like calculate a route to my house on this map, it would be pretty difficult. I mean, how would you work out the house was there compared with, say, my neighbour's house, which looks entirely different? So it'd be quite difficult to pick out and so on. So these two ways of representing the data, either by a set of coordinates or by an array of dots where you store the colour value of each of the dot, give you a different sort of information, and they are useful for different things. And there's a great deal of power in adding the two together, as we can see here in Google Maps. So environmental layers, as I've used them, are rasters. So they consist of an array of dots. In this case, each dot represents a kilometre square. This one is mean altitude from a digital elevation model. This is freely available from the Ordnance Survey open data. It gives the altitude of every point across the British Isles every 50 metres. I've simply averaged these for a 1 kilometre square. And so each dot is a 1 kilometre square, and the value displayed here as a colour is the height of the mean height of the ground in that 1 kilometre square. Similarly, this is the CEH, Centre for Ecology and Hydrology land cover maps. Again, freely available. You can download them from CEH's website. And in this case, each dot, each 1 kilometre square, represents the area of that 1 kilometre square in hectares that's covered by one of 21 land cover types. And this one is arable and horticulture. So you can see that east of England and to some extent the east of Scotland is where most of the arable land is concentrated. So where I live around here, you'll really struggle to find a cow or a sheep, but you'll find pretty much wall-to-wall coverage of wheat fields and this time of year oilseed rape fields and so on. And this information tells you that. Notice it doesn't cover the Isle of Man. Bioclimatic variables. Met Office has gridded weather data. This is information like average, minimum and maximum daily temperatures, rainfall, hours of sunshine, wind speed, cloud cover. Some of these variables, I think in the case of some of the temperature variables, go back to the late 19th century. pretty much all of them are available from 1960 onwards. And again, you can download them, Hadley UK gridded data sets, by getting hold of the daily average, minimum, maximum temperatures. I can then turn these into what are known as bioclimatic variables, 19 variables that are reckoned to give good proxies for the sort of things that matter to biological species. Things like the maximum temperature of the warmest month, the minimum temperature in the wettest quarter, those sorts of things. And finally, soils. So this is data from the European Soil Database. Again, freely download it. There's the URL. And this one shows the dominant soil type in 1 kilometre squares across Britain. So each of these colours represents, I can't remember how many different soil types it is, but it's somewhere in the 40s. And clearly southern England is fairly different from northwest Scotland in terms of the soil types that cover it. So you can probably guess these shades of green represent upland peat types. So I have a whole load of different layers, and I think that comes to about 42, and we'll see some of them later on.
Stuart, do you include hard geology as well amongst those?
Nope.
No.
It's well, I could probably find a source to get hold of it, but I think soil and drift is far more important than hard geology, really. Although it's not something I've specifically tried out.
I think it might well be worth looking at, but that's for another time.
Yeah.
Okay, thanks.
There's a whole load of different ways of conducting species distribution analysis that have been suggested over the years, and these go back to 1960s, 1970s. The first and most basic, which actually came out of work in, I believe it was Swansea in about the 1960s, and you still find bioclimatic envelopes talked about, was if you take the places where you know a species occurs and you look at things like minimum and maximum rainfall, what range of rainfalls encompass that species distribution. You can then take the overall rainfall map and draw a circle around the areas that encompass that range. If you do the same for temperature, you'll get another circle. And where those circles overlap, that's the potential envelope in which that species could occur. And then add, another factor and another factor, and you get more and more intersecting circles, which hopefully narrow down the spots in which a species could occur. And this was applied worldwide, particularly to snails, a long way back. And you can still find computer programs and packages that will do this sort of stuff, extending it beyond just bioclimatic, but also to habitat factors and things. Ecological niche factor analysis. This is something that is very popular, particularly with large mammal people, particularly for research on large mammals in Africa. It's been very heavily used. And this is an extension of factor analysis. Factor analysis is a method whereby if you assume that variables are normally distributed, you can describe a series of observations as points in a multi-dimensional space where each dimension is represented as a normal distribution along one of these variable axes. And then you can try and cluster these points in hyperdimensional space. So if you take the known places where hunting dogs occur or whatever, try and express them in terms of things like temperature, altitude, land cover, and so on. You can then try to cluster those points in hyperdimensional space and therefore work out what the sort of niches of this species are. The big problem with it is that a lot of environmental variables don't readily, are not normally distributed. And whilst you can apply mathematics to try to normalize them, it really doesn't work very well, particularly for things like land cover, because if you think about land cover, if you think about a land cover like salt marsh, across Great Britain, that will have an awful lot of squares where it's zero. And trying to normalize a distribution that has a large number of zero values is very difficult. So I've never found ENFO works very well for the sort of stuff I've looked at. Next, we get regression-based models. You can obviously think if you ran a series of malaise traps along some environmental gradient. So you run them at a series of different altitudes going from the coast up to the mountain top. And you look at the proportion of the catch that was of Episurfus bultiatus in each of those malaise traps, and then try to do linear regression against factors like altitude or temperature, which tends to be strongly correlated with altitude, and so on and so forth. General linear models are a way of generalizing linear regression into areas where you're no longer assuming normal distributions. You can assume other sorts of distributions and so on. And general additive models allow you to also decide that the shape of the relationship between a variable and whatever you're measuring is no longer linear, but follows some other arbitrary curve described by something called a spline function. Generalized additive models are very popular these days, and you see them used a lot. Generally, these methods require some form of presence-absence data. You need both presences for your species and some decent idea of places where it doesn't occur. So they tend to be quite strongly used where you've got things like trapping data. So if you had a grid of pitfalls, and you can look at those pitfalls and say, what's the count of the species in each pitfall, which could be naught up to 100, or you could actually express those traps as the species was present in it or was absent in it. It was never caught. They have some advantages in that these sorts of models, you can do all sorts of regression statistics where you can try to partition the variability amongst your different environmental variables. You can get some idea of what's driving the distribution. You can partition that up and try to get partial correlations. So how much is each factor affecting the distribution and so on, which can be somewhat difficult in this next category of models. The final category is really machine learning models, and these have come along particularly since 2000, and I think have taken off in a big way with, they take a lot of computing power, and until particularly PCs had got powerful enough to run this sort of thing, allowing a lot more people to use them, they didn't really get much traction. So I would say really from probably the 2010s onwards, these have become the sort of dominant area. And really here, various machine learning techniques as pretty much black boxes to try and develop a relationship you can think of as pattern matching between the places the species occurs and the environmental variables. And one of their problems is they often are rather black boxy, that it's difficult to know what's going on inside them. So actually deciding what is driving the model is quite difficult. There are ways of tackling it empirically to try to um, tease out what the most important environmental variables are, but they're a bit more indirect.
Sorry, Stuart. I think your microphone might have changed slightly there.
Okay, is that? Am I going very quiet or something?
Now you're
Yeah.
better then.
Yeah,
Okay.
it's better now.
Great, fine.
Thanks.
Okay, so GARP, general algorithm for rule set production, uses a genetic should be genetic, not generic, genetic algorithm, which attempts to use something like a natural selection system. You assign values to all your parameters in the model randomly. You fit a whole load of models. You choose the ones that fit best. You then get rid of all the poorly fitting models. You mutate the ones that worked well by tweaking the algorithm, tweaking the parameters slightly using some random process. Generate a new set of models from those and go on and try to use a sort of natural selection to cull out the ones that don't work. We're going to come back. Maximum entropy is the one I'm going to demonstrate, Maxent. We have decision tree based things, decision trees leading to random forests of trees, boosted regression trees, and finally neural networks and things like support vector machines and artificial neural networks. I could vaguely have a go at trying to explain maximum entropy in random forests, but I have no clue how a support vector machine works, I'm afraid. I can fit them, but I have no idea what's going on under the hood in a support vector machine. Okay, so I mentioned Maxent. Maxent is the piece of software I've used most often. This is maximum entropy modeling. Again, it's free and open source software. You can go and download it, no problems. Its big advantage is it's fast and it doesn't require an especially powerful computer to run. Some of these techniques take a lot of computing power and take a long time to fit. Maxent's not bad. It does require that your machine has Java, but it's uncommon to find a computer that doesn't have Java. I've given here two pretty essential references. Phillips, Dudick and Shapir's paper, which gets constantly updated as new versions are produced, and a really good paper by Jane Ellis and Phillips, who's the author of Maxent and others, on a explanation of Maxent for ecologists. And if anybody wants to get into this, I would really recommend reading that paper. It is very, very useful. I certainly I keep a PDF available and refer to it quite often. So to run Maxent, I use these environmental layers, which are 1 kilometre square rasters, stored in a simple text form, Esri ASCII grid format, which Maxent can take. And there's another format Maxent can also take. I supply species data as a simple CSV file with the names, species name, and XY coordinates in terms of kilometres of the OS national grid. One limitation of Maxent is it wants the data in a format where all the rows relating to a given species are grouped together. That's pretty straightforward. You simply sort the file by the species name, no problem. Final note is Maxent, certainly the latest versions of Maxent, will do some parallel processing if you've got a multiple core computer. And that does speed it up a bit, but it really doesn't make a very big difference. let's give it a go and see what it looks like. So what I was planning to do was first to talk you through actually preparing the data, then run Maxent for, I've been mentioning crosstalk and arcuatum, so we'll use that, and then actually looking at the results and evaluating the results. So to start with, what I'll do is talk you through generating the data, and this follows on a bit from the last session we did. So if those of you who saw the last talk about trends will know that the tool I use for most of this is R. R is an open source data manipulation and statistical programming language, and I'm using this piece of software called RStudio, which is an integrated development environment for R. And those two together are the tools I use for almost all of this. If you wanted to see this in a bit more detail, go back to the previous talk, which I believe was recorded and I believe is available online on Diptris Forum, I think. You'll see some more detail. But if I load an example, I can show you what's going on here. So again, harking back to my previous talk on R, R uses a basic core, which is relatively simple and largely does the interpretation of the commands you give it and sends them off to functions. And functions can be supplied in packages, which are external chunks of code which add functions to R. And there are tens of thousands of packages out there to do almost anything imaginable. And here I'm using a couple of packages, one called data table, which is used for manipulating large sets of data. And the Hoverfly recording scheme these days is a pretty large set of data. We're dealing with 1.75 million records. So manipulating data on that scale can become quite slow. Data table is very fast and also takes advantage of parallel processing on multiple CPUs if your machine's got them. So it speeds things up a lot. The other package I'm loading here is called Lubridate, and it's date processing functions. Things like the one we'll use here is getting the year from a date. So straightforward. So we'll load those two packages. We'll set our working directory, which is just where I keep my R stuff, and we'll clear out variable. We haven't got any variables in our environment, but it's always good to clean it out to start with. And I will set a couple of variables for a start date and an end date. Those are when I'm going to start and finish this analysis. I've actually put 23 there, so I'll change that to 22, which is what I've been saying so far. Now, going to load the Hoverfly recording scheme data, and let's just have a quick look. Hover stats is where I keep my, so it's the one we set to. And in my data temp directory, these are just dumps from the Hoverfly recording scheme data. And you've got species, date, and grid reference basically for each record. And there are just under 1.5 million records in that file. So that file is 78 megabytes of data. So we'll load that. And as I said, data table is quite good. Loads that data pretty quickly. There I've got various date variables, and you can see data table has recognized their date, but I actually want the year so I can work out my year range. So I'm using this Lubridate package to extract the year from this start date variable. And so there we go. And we should find now we've got a year variable in there. There we go. Now, the second thing I need to do is get the 1 kilometre squares and get their XY coordinates. Because if I remember, Maxent wants species name, XY coordinates, the presences. So the first thing I'm going to do is I've got this grid variable, which is text. So if we just have a quick look at some of the data, you'll see this is what the grid looks like. It's the OS grid, and it can be 6-figure grid references, 4-figure grid references, occasionally 10 kilometre square, 2-figure references, or occasionally these things from GPS, which give it down to the nearest metre, I think, in that case. So I'm just going to clean that up a bit by making sure everything's in uppercase and there are no spaces in the grid reference, because sometimes people put spaces between the letters and the figures or whatever. They should have been cleaned up before they went into the database, but we'll just make sure. And secondly, I want to extract the 1 kilometre squares. Now, if we go back to here, I can try and explain this a little bit. So I popped out into the garden and actually used the GPS on my phone to get the grid reference. That's grid reference in my garden, and that's what GPS reported. So that is a 10-meter grid square, an 8-figure grid reference. Clearly, that can be simplified to a 6-figure or a 4-figure or a 2-figure, a 10 kilometre square, 1 kilometre square, 100-metre square, or this 10-metre square. I want this format, so I want to extract that out of that. So clearly, the first thing I can do is take the first 4 characters, because that's always going to be the first part of that 1 kilometre square. So what we're doing here is taking a substring of the grid starting at 1 and ending in 4. So it's those 4 figures. I then need the 2 figures representing the Y part of the 1 kilometre square reference, which are those 2 or those 2 or those 2. So I've got to work out where they come. So if we just do 1, 2, put a space in. In that case, they're going to start at 5. 1, 2, 3, 4, 5. That's going to be the first figure we want. In that case, they're going to start at 6. In that case, they're going to start at digit 7. So what I'm doing here is I'm taking the number of characters in the grid, which is going to be 4, 6, 8, 10. I'm taking 2 off, which are the 2 letters, and I'm dividing by 2. So that's giving me the number of figures, which would be 1 in that case, 2 in that case, 3 in that case, 4 in that case. So length 10 minus 2 is 8, divided by 2 is 4. And then you can see in each case, that was 2 figures, that was 3 figures, sorry, and that was 4 figures. You can see that we need to add 3 to the number of figures to get where we need to start. So the number of characters in the grid minus 2 divided by 2 and add 3 to it, and that gives us our starting figure. And I want to do this, and I just want that figure and the next one on. So I then need to do the same again, and this time add 4 to it. And when I stick those bits of string together, I want to use nothing as a separator. just stick them together without a separator. So if I do that, I should have the 1 kilometre squares. And if we look at this, we can see 1, 2, 3, 6 is what we've got. In this case, with 4 figures, 6, 7, 6, 4, which is what we've got. We go down to this one, which was a 10 kilometre square. You can't get a 1 kilometre out of a 10 kilometre square. So this has produced rubbish. SJ288 with only 3 digits is not a proper square. So it's produced something, but it makes no sense. And in this case, where we've got loads of things, we've got 14, and then I think it'll be 7, yes, 7, 8 will be the start of the Y figures. So they're working correctly, except we need to do something about these cases where we had a 10 kilometre square. So what I'm going to do next here is filter these records to the ones that are within our year range. So the year is greater than equal the start year, and the year is less than equal the end year. And then I'm going to check that this 1 kilometre is actually what we want, which is two letters followed by 4 numbers. So this says I want to match them, and this grep is regular expression matching. I want the start of the string, which is what this little caret character says, followed by 2 letters, followed by 4 digits, followed by the end of the string, which is what this $sign represents. And I'll only accept cases where 1 kilometre square matches that. So that's cut us down to 830,000 records. I've now got a file in my data, which is the 1 kilometre square coordinates of every square on land in the British Isles. So there's the 1 km square, and there's the kilometre coordinate, centre coordinate of that square. And maybe one day we can have another one of these talks where I can show you how I get things like outlines and these lists of squares and things, because that's quite interesting as well. So we'll load that. And there we've got the coordinates, 1 kilometre squares with their XY coordinates. And we're simply going to join them. And what this is doing is joining coordinates and observations on this field called 1 km, which is present in both of these data tables. So if we do that, we'll find that our observations now have XY coordinates in there. Now, what I actually want are the unique set of XY coordinates for each species. So I'm going to extract the species X and Y from each of my observations. I'm going to get just the unique ones, and I'm going to knock out any where anything's missing. So if there are any 1 km squares for which we didn't get coordinates, it'll drop them. And I want them sorted by species, so I've put order species in there. So what I should end up with is this data table called species XY, which has species, XY coordinates, and should be sorted by species. So there we go. Bacca, Chrysotoxinum, Helophilus, so on and so forth. And all I need to do now is save that to a file. So I'm going to stick it in this temporary file here. I'll call it species XY, and I'll put the start and end year in the name so I know what was in this file. So that generates the file name, so we can have a look at it. It's going to be called Species XY 2010 to 2022, and it's going to be in temp species, and let's write it. And there we go. So if we go and have a look at that, it was in C temp, and there we have it. 2010 to 2020, and the first line of the file has the column name, species and XY, and then we have species name, XY coordinate, and they are sorted by species name, which is what Maxent wants. So now we're ready to actually, before I do that, we'll just have a quick look. Here's a directory which has environmental layers in it. So if we look at one of these, this was the mean altitude. This is what it looks like. It has a bit of header information, and then it's got a value for each 1 kilometre square. And the -99s mean no data. So these are the C. So if we scroll down a bit and scroll across, we'll see here the altitude for various 1 kilometre squares across the north of Scotland, because this is near the beginning of the file. And the header is telling it has 660 rows across. It has 1,220 rows north to south, and the bottom left corner is at 00, which is somewhere off the Isles of Scilly. And the cell size is 1. So these represent 1 kilometre squares in this particular case. So pretty simple text format. And you can see that that's about 6 megabytes of data. And we've got here mean altitude, the amount of south-facing slopes, a measure of the roughness of the ground, the dominant soil categories, the various land cover categories, and the various bioclimatic variables. And there are 42 files in that directory. So that's the data we have available to us. So let's go ahead and look at Maxent. Maxent, there it is. So if you download it from the web, this is what you'll get. You'll get a file, a zip file, which has a batch file, a Java file, a jar file, an SH file, which is for Linux or Mac to run it, and a text file, which gives you some basic information about how you use it and so on. And it's the BAT and the jar file we want to use on Windows. And if we look in the BAT file, we'll find it's simply got a Java command to run Maxent, and it sets the amount of memory. And you can easily change that for the amount of memory you want. So we'll simply double-click that batch file to run it. And there we are. We are running Maxent version 3.4.4, which is the latest version. And it's got two things it's asking for here, the samples and the environmental layer. So our samples were in C, temp, and we'll use this 2010 to 2022. And there we are, it's found all the species. Now, I'm not going to run all of them, so I'll deselect all, and I'll scroll down and I'll find the species we were going to look at, which was Chrysotoxinum arcuatum I was going to run. There we go. Secondly, we want the environmental layers. So that was in D, sorry, it was in E. Hoverstats. I have this problem with Java. I often find it difficult to get the blooming file reader to actually open directories. It's a pain. And we want the layers. Open that and it will come up with the 42 layer files. Now you can see here, it's assuming they are continuous variables. There's two sorts of variables, continuous and categorical. So continuous, things like altitude. So that would be the mean altitude in meters, and it could be anything between 1 and what about 3,000 to 4,000 in Britain. And the other type of variable are categorical variables, where things like the soil type, 1 and 2, 2 isn't bigger than 1, it's just different from 1. They're categories, they're not continuous variables. And I name my categorical variables starting with cat, just to remind me. And you've got to tell Maxent that's a categorical variable, which we can do like that. We also need an output directory it's going to write to, and so I will use Maxent. Come on, it always does this to me, results. I don't want any of these things, but I do want to change some settings. I'm going to use 50% of my observations to train the model, and I'm going to use 50% of them to evaluate the model, and we'll come to that in a minute. Otherwise, I think I can use defaults, so I'll just leave everything alone. What I will do is tell it can use more threads, because that might speed things up a little bit. So I've just changed a couple of settings, and we should be good to go now. So we're accepting all the defaults except just those two exceptions, which is 50% of the observations for testing and use multiple threads. So if I run it, it's telling me that some of my observations are missing environmental data. Now, I'd expect that, because if you remember, some of these environmental data sets didn't cover the Isle of Man. So you would therefore expect them to be missing values. So that's expected. So I'll just say suppress similar warnings to just get rid of all those messages. And now it'll go ahead and do it. It's now fitting the model. Gain, which is going up, is a measure of, it's from signal processing. It's a sort of measure of amplification, how much more information the model is giving us than is available in the original random trial of the data. So the bigger that gets, the better the model's likely to be. And now it's done. So it's done that. Not particularly, doesn't take particularly long. So let's have a quick look at it. It was in E, Hoverstats, Maxent, results. And there we've got it. And it conveniently writes us an HTML file, which will let us look at the results. And we'll come back to this in just a minute. It's giving us some estimation of the importance of the various variables, and it's telling us it used 304 present records for training, 303 for testing, and it used 10,000 background points to determine the distribution. And it's given us this estimate of the likelihood of species occurring in the 1 kilometre squares. Now, again, we'll go back into R and we'll have a look at those results. So I don't save that. File, recent files. We'll visualize it. This is a raster. This is a raster map. So I'm using an R package called raster, a very powerful package for handling raster data. We'll load that. As exactly the same as we saw before. So I'll load my raster now. It's loaded this raster, and I can simply say plot R. And there's my potential distribution of Chrysotoxinum arcuatum. The darker green colours, the higher chance of the species occurring in the square as far as the model is predicting, and down to the sort of beige colour, which is no chance. Now, we could improve that a little bit. I'll load here an outline of Great Britain without the Isle of Man because Isle of Man's missing from this data. I'll plot this again, but this time I'll leave the axes off and I'll use a different color scheme. And then I'll plot this outline I loaded around it. So that's, you know, a reasonably nice visualization of what Maxent has produced. So you can see R, and particularly using the raster package, can deal with this really nicely. And obviously we could go on to lots of other analysis, that sort of thing. The other thing you can do here is this is 1 kilometre squares, and there are about 750,000 of them. So it takes a while to plot. So aggregating them up to, say, 2 kilometre squares can be quite useful because it reduces the number of squares by a factor of 4, you know, 2 by 2. And so it'll improve the plotting speed and you'll be hard put to see any difference in the results. So if I run that again, using this time aggregation and plot it again, I think you'll be hard pressed to see any difference. So I often aggregate a little bit just to improve the plotting speed. Okay, so let's go back and look at the last little bit of this. Okay, so we ran a model, we got a model. Is it any good? So an important thing is to evaluate that model and determine whether it's any good or not. The result we got out is this thing called a logistic probability for each 1 kilometre square, which are here shown as colours. So the redder, the higher, the bluer, the lower. We can turn this into a presence-absence prediction simply by applying a threshold. So, for example, we could say if that value was greater than 0.5, we'll say it occurs in that 1 kilometre square. And if it's less than 0.5, we'll say it didn't occur in that square. And once we've got this presence-absence prediction from the model, we can compare that with actual data. And so we can determine how well that prediction fits the data. And what we end up with is the situation where we've got our observations shown here as the yellow dots, and we've got the model predicts presence using some threshold, and that's shown as the blue squares. And there are 4 possible outcomes. It was observed and the model predicted it. It was observed, but the model didn't predict it. We've got a yellow blob here with no blue background. It was not observed, but the model predicted it should be. The result we want, really. And finally, it was not observed and the model predicts it shouldn't be there. So there are 4 possible outcomes. We can readily count those 4 possible groups of squares. And what we end up with is this thing called the matrix of confusion, where we've got our observations, whether it was observed in a 1 kilometre square or not, and our model prediction of presence or absence. Remember, this is for a particular threshold of that logistic probability. And the 4 possible outcomes referred to as true presence, false negative, and true negative. In this case, you can see of our 131 observations, 127 of them were where the model predicted presence, and 4 of them were where the model predicted absence. And of our 10,000 background points that we randomly selected to use for the model, it suggested presence in 1,000 of them and absence in 9,000 of them. So from this, we can get 2 important results. The true positive rate is of these 131 observations, how many did the model correctly predict, which in this case is 127 out of 131, which is 97% essentially. And the other one is the false positive rate, which of these 10,000 background points, what proportion of them did the model suggest were presences. So that's 1,005 out of 10,000, so that's 10%. What we can then do is repeat this calculation for each possible threshold between 0 and 1, between 1 and 100%. And for each of these points, so each of these represents a threshold, we can work out the true positive rate and the false positive rate, and we can plot them, and we get this curve, which is known as the receiver operating characteristic, or ROC curve. And we can work out the area under the curve, which is this area here. And that's known as the AUC, the area under the curve. Now, theoretically, if our model was absolutely perfect and always got it right, then the curve would look like this and the area under it would be 1. If we simply threw a coin with no extra information, but just flipped a coin for every occurrence, we'd get this diagonal line, and the area under the curve would be 0.5. So more that the curve pushes into this corner of the plot, the better the prediction across the whole range of thresholds. It is possible to get a curve that actually comes below that 0.5, and that's a perverse model that's essentially predicting absence where the species is present and presence where it's absent. And you do very occasionally get that. So as a rule of thumb, and this is very widely used in the medical field and so on, generally people regard about 0.8 as an acceptable model, 0.85 as a reasonably good model, and 0.95 as a very good model. And again, the general rule of thumb, if it's 0.95 or better, then you probably cheated and biased it in some way. So what we tend to do is randomly split the occurrences into a training set and testing set. We fit the model using the training set, and we evaluate the model using the withheld data as, in some sense, independent. It's not truly independent data because it comes from the same source, but it's in some sense more independent. And what we pay attention to is the testing results, not the training results, because we'd hope the training curve would be pretty high if the model fitted successfully. So in our case this time, the training AUC was 0.933, but the test AUC was 0.887. So going back to our previous slide, we would say that's a pretty good model, really, I think. And that really is what I wanted to do here. I wanted to show you the results, show you the process, and say a little bit about the evaluation process. And really, there's quite a lot more to be said about the problems with these sorts of systems, how they can be mitigated, some small stuff around this evaluation process and so on. And that's what I'd like to cover next time in a couple of weeks. So I will hand back to Roger in that case.
All right, now I've just unmuted myself. Right, well, thank you very much indeed for that, Stuart. That certainly got me thinking. I hope it's got everyone else thinking. Any questions, any thoughts?
What I'll do is stop sharing my screen.
That would be good, yeah.
Yeah.
Right.
Done. Silence.
I've got a question. So it was really great. Thank you very much, Stuart. Excellent guide through that process of modelling. You may have covered this in your previous talk, which I'm sorry I didn't catch, but do you pre-filter the HRS data for like recorders with a certain number of records or that kind of thing? Or is it all records?
Well, we do have in the data quite a chunk of records that we question for one reason or another, and those are not included in this. But in this particular case, I have simply taken all the records that we have basically decided look as if they are reasonable, and so are not flagged as questioned within the database. When we get back to talking about biases and things that might affect the modelling, then that's the sort of thing we might consider. And certainly in the last talk about trends, the fact that since about 2013, when the Facebook group started, there has been a considerable change in the records we're receiving. So they've become dominated by photographs of a few common species. But if you're looking at the places where Chrysotoxinum arcuatum occurs, in this example, that really doesn't affect anything. I mean, the fact that 50% of our records now are of Episurfus bultiatus, it doesn't take anything away from the fact that where Chrysotoxinum arcuatum has been recorded, however it is, that is a point of presence we can use for this model. So I don't think that's too much of an issue for this particular purpose, but it does become quite important when you start to look at some other things like trends in frequency of the species, which is what we discussed in the previous talk.
Great, thank you.
Make a couple of comments.
Yep.
The statistical modeling that you could do, you mentioned GLMs, GAMs.
Yep.
I think for hoverfly data, probably occupancy models are more useful. And they correctly model the varying efforts, they correctly model the fact that absence doesn't necessarily mean absence.
Yep.
And point process models also offer options that address this. So I think in the future, there'll be more development of those types of models.
Yes, I would agree with you. And obviously, I've played a fair amount with the stuff that your colleague produced and so on. And I've tried my own general additive modeling, and that can be quite impressive. It does, I would say, it does take an awful lot longer in general to simply run the models. So for a demonstration like this, it's quite tricky. Some of the processes for the next talk take a long time to run. So what I've done there is I've recorded screen movies and edited them and drastically speeded up the bits that take 20 minutes and that sort of thing. So it would be quite possible to try and talk more about that sort of thing. But, you know, in doing a demo like this, Maxent has the advantage of speed in trying to present the concept.
Yes, definitely.
Yeah.
Yes, it was impressive that it would run just while we were watching it in seconds.
Yeah.
One question on your terminology.
Yeah.
You were describing false positives as the ones where presence wasn't recorded, but was predicted.
Yeah.
I would have called those false negatives because they're not recorded there, but the model says they should be there.
Well, that's just generally what is used for rock analysis. And I think it comes largely from medical diagnosis, doesn't it? The sort of false positive, false negatives thing.
It does, but I would have reversed the labels. Wouldn't make any difference. I'd just reverse the labels.
Fair enough. Yeah. I'd need to look
your
back at my slide.
context matters considerably. Are you taking it from the point of view of presence or absence? And you need to be absolutely clear about that, which is what you're both saying. One's taken from the point of positive presence, and the other one's taking it from negative is the absence. Yeah? It's just how you look.
It's a question of whether Yeah, it's a question of whether you take the model predictions
Correct.
as the force or the observations as force.
So you need to be
And
clear
I would have
about that.
said,
That's all right. There's no argument about it. You just need to be clear what
no.
you're doing.
I think it's false
So
positive.
is it
It's
clear
false
the way you label
positive.
it? Doesn't matter.
Yeah, it's false positive in the sense that it was not observed, but the model predicts it should be there.
I personally, from my point of view, I'm going to go with Steve's approach on it. Because if we're evaluating, has the model predicted what's happening, therefore what we see in the cell, is it a false negative from the point of view of the recording scheme?
Yeah.
But yeah, it's a,
Yeah.
do you start from A or do you start from B?
Yeah. Yeah. Well, I've just followed the terminology that is normally used in, if you go and look at sources to how to calculate a rock curve, that is what the terminology tends to be used. Yeah.
That may be because a negative, observed negative is negative in that context.
Yeah.
But in our context, a negative means you haven't recorded it.
Yes.
Doesn't mean it's necessarily absent. So
Yeah.
I think that's the reason it looks a bit odd to us.
Fair enough. Yeah.
Quick question on your conversion of OS grid references to numerical. Do you have a function that does that, or are you doing a lookup? Because I've got a function that works across all channel islands included if you want it.
Yes, I've got functions that do it. Again, it's easier to understand in terms of doing this sort of demonstration, and it's a hell of a lot faster.
It's okay. I just couldn't find one when I wanted one, so I had to make it. I'm trying to save people that trouble.
Well, I've produced my own, so I have my own stuff. But there is actually a package produced by CEH called Sparta, which has functions to do XY to grid and grid to XY. As I said, it would be nice possibly one day to look at some of this grid handling and that sort of thing. Because if you actually start doing things like GB outlines and so on, the source of readily available free geographic data is mostly in WGS84 lat long format. The source of European data, like the European Soils Database, is in a grid system called Lambert equal azimuth. And so we do end up doing conversions between grid systems quite a lot. And raster, coupled with another package called RGDAL, are very, very good at those sorts of things. And it's something that people wanting to manipulate biological records would very much benefit from knowing about, because it's a point that comes up quite a lot, converting things between different spatial projection systems.
Can I ask, you mentioned the 42 environmental variables, and I noticed it went through quite quickly. You had a table which seemed to suggest that some were more important than others for the particular species. Does the model then bias the result to those that seem to be more important?
The model is trying to find, as I understand it, a pattern of environmental variables that are the most different between those points of presence and a random selection across the whole of that geographical area. It will then use various techniques to try and give you some empirical idea about how much those various layers contribute to that difference. And that's what we get. I mean, if you'd like to look at that in a little more detail, I could show you those. So if I go back to sharing the screen and go back into that. So if we,
Yeah,
it's showing here percent contribution, which is an empirical estimate to how much these different layers contributed. And you'll see there's a whole load of them that contributed nothing or very little indeed. But the ones that contributed most were in fact woodland cover, which is coniferous and broadleaved. Arable horticulture, in fact, one of the things it can do is it can estimate the relationship between these variables. And you'll find for coniferous woodland and broadleaf woodland, it's positive. And for arable and horticulture, it's negative. In other words, the coverage of arable land is a good way of telling you where this species isn't. It's also finding temperature, rainfall, and soil type. And really, once you get below about, this 5% level, you're getting into pretty minor variables that don't have much impact. So one of the things we'll come back to next time is simplifying, selecting the variables, reducing the variables. Just one of the problems is these variables can be highly correlated. So in general, altitude and temperature are going to be very correlated, for example. The researchers suggested this doesn't affect Maxent to any important extent, but it's one of the things you might want to do is to actually reduce your data set down to the most important variables. And clearly that can be helpful in looking at what sort of features you might be interested in if you're researching that species. But we could guess from what we know of the biology of Chrysotoxinum arcuatum, genus Chrysotoxinum are thought to be feeders on ant-attended root aphids in ants' nests. So they are in places where there are likely to be good populations of ants. And that will tend to be quite warm microclimates. So hence, warm conditions are probably quite good. Well-draining soils are quite likely to be good, which is where you'd think soil might come in. And you nearly always find the adults along woodland edges and scrubby places and things like that, which is where woodland cover and particularly woodland edge might be an important factor, and where having large areas of open arable land would be a negative factor where you wouldn't expect the species. So this does make sort of sense biologically in relation to that species. And you might actually want to hone down your set of variables based on your knowledge of the horticology of that species, if you have any knowledge of it.
And can I ask one follow-up question?
Yep.
You're in the example you gave, one of the places where it was occurring but it wasn't predicted, I noticed had very limited land within the monad. And if you're averaging, then you're possibly not. That's not a very accurate prediction.
Could be, yeah. I mean, if you get into species which are related to very habitats which generally have very small areas, then this sort of land cover data at that scale possibly is not very useful. And the ITE land cover data is actually at basis a categorization of 25 meter squares. So you could model down to that resolution.
I'm not suggesting that it's really a big problem, but it is where you only have sort of 3% that was where it was, I think it was 3%, where it was not expected, but was occurring. And if some of those you got a lot of sea and therefore clearly not got coniferous woodland.
Yeah. Yeah.
Except possibly there was coniferous woodland sort of on the coast, which I
Yeah.
think there is in that particular area.
Yeah. Yes, I think edge effects are always a problem in these sorts of systems, and I'm not quite sure what you do about that, really.
No, I'm not. I recognise that.
Yeah.
Thank you.
Right. Does it look like we've covered everything? No one any more questions? Right. Well, Stuart, thank you very much indeed. That was very comprehensive. So hopefully we'll see a similar group in 2 weeks' time, and Stuart can continue the adventure, as it were. It's quite interesting seeing who was here. It's quite a small part of the UK Hoverflies group, and very interestingly, various people from other areas of interest, which is great, because we're actually touching a broader spectrum of people. But amongst the Hoverfly people, perhaps it's worth just encouraging people to log in, because some of the stuff that Stuart will be doing next time, I suspect, will start to interest people, and it has quite a bearing on the sort of data that we're trying to encourage the collection of. So, right, I look forward to seeing everyone in 2 weeks' time.
Thanks, Stuart.
Okay,
Really
I'll
good.
stop recording.